Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearsonetic Cell International A-Level, Biology Unit 5 for January 2024. Let's begin with the first question. Question 1. Aerobic respiration is made up of several stages, which is the correct order of stages of respiration required for the complete oxidation of glucose. Respiration goes through glycolysis, the link reactions, and the Krebs cycle, so the answer should be B. Next, they say write a balanced equation for the complete oxidation of glucose in aerobic respiration. During aerobic respiration, glucose is going to be reacted with oxygen. We produce 6 carbon dioxide molecules and 6 water molecules, so this should be the equation. And next, they say the equation shows the aerobic respiration of a lipid, which is this one here. They say which is the respiratory quotient when the lipid is used in aerobic respiration. Respiratory quotient is volume of carbon dioxide released divided by the volume of oxygen used. Since here we have only 2 gases, the volume ratio is going to be proportional to the mole ratio. So RQ is going to equal to the moles of CO2 released divided by the moles of oxygen used, which is going to be 36 divided by 51, giving us 0.71, and therefore B is the answer. Next, they say the complete oxidation of one molecule of this lipid produces 122 molecules of ATP. One molecule of this lipid contains 12,244 kilojoules of energy. One molecule of ATP yields 30.51 kilojoules of the energy. Calculate the percentage of energy in this lipid molecule that can be converted to energy in ATP. They say give your answer to 3 significant figures. From the information given, 1 mole of the lipid contains this amount of energy, and this contains 122 ATP molecules, whereby each ATP molecule is going to yield this amount. That means the amount of energy yielded by the 122 molecules of ATP is going to be 122 times 30.51, which is this one here. So the percentage is going to be that divided by the amount of energy in the lipid times 100, giving us 30.4%, and that is the answer here. In question 2, impulses are transmitted along nerves as a series of action potentials. An action potential can be split into 4 main stages. There is depolarization, repolarization, hyperpolarization, as well as the resting state. They say which row shows the events that happen during depolarization of a neuron. During depolarization, the sodium ion channels are going to open and sodium is going to enter inside the neuron, so the membrane potential is going to increase. The answer should be D. And next is explain what happens during repolarization in a neuron. During repolarization, the sodium ion channels are going to be closed, meaning the sodium ions are no longer entering into the neuron. And the potassium ion channels are going to be open, meaning potassium ions are leaving, so the membrane potential inside the neuron is going to decrease in comparison to the outside. So I say during repolarization, the voltage-gated sodium ion channels close, so the influx of sodium ions stops. The voltage-gated potassium ion channels are open, and potassium ions move out of the cell. This causes the membrane potential to become negative in comparison to the outside. The photograph is a cross-section of part of a sciatic nerve showing the neuron. So we can see the single neurons, which are very small in the whole nerve. And then they say, calculate the magnification of this photograph. They want you to give your answer in standard form. I used this scale bar here, and the image length was 9.5 millimeters. Since magnification is going to be image length divided by the actual length, the actual length was given to us as 50 micrometers, so I converted this into micrometer. It's going to be 9.5 times 10 power 3 micrometer. So dividing, I got 190, which is 1.9 times 10 power 2, since our answer should be in standard form. So that is the magnification. And next I say name a type of microscope that will be used to obtain this image. This is going to be a light microscope because the magnification is not that big. Explain how myelinated neurons enable a greater speed of transmission in a neuron. If neurons are not myelinated, conduction has to occur along the whole axon. However, if myelin is present, then conduction only occurs at the nodes of Ranvier, so there is going to be faster conduction. So I said, in the myelinated areas of the axon membrane, ions like potassium and sodium cannot cross, so conduction only occurs at the nodes of Ranvier, which is saltatory conduction, and this occurs as the impulse appears to jump from one node of Ranvier to the next, speeding up the conduction process. Also, myelin acts as a good insulator for the neuron, preventing short-circuiting and loss of membrane potential. So this brings us to the end of question 2. Let's continue to question 3. In question 3, the photograph shows a roundworm, which they call the C. elegans. They've given us the magnification as 200. They say the roundworms are simple animals with a nervous system consisting of 302 neurons. The roundworm can respond to stimuli with reflex actions. A reflex action is a rapid involuntary movement in response to a stimulus that involves receptors, which receptors detect a change in light intensity and stimulate an action potential. Because they say it responds to light intensity, these should be photoreceptors, and the answer should be D. Baroreceptors respond to pressure, chemoreceptors to chemical changes, osmoreceptors respond to iron and water concentration within the body. A reflex act carries a nerve impulse from receptor in the skin to the central nervous system in a human. Describe the structures that a nerve impulse passes through from the skin to the central nervous system. In the skin, the receptor is going to detect a stimulus and convert it to a nerve impulse. This impulse is going to be transmitted to the sensory neuron, and then the sensory neuron is going to synapse with a relay neuron. Since they say this is going to be a reflex arch, so we have to talk about the dorsal root, because the sensory neuron is going to be in the dorsal root. The impulse is going to be carried through the sensory neuron to the central nervous system. So I said after generation of an impulse in the receptor cells, the receptor synapses with the sensory neuron. The impulse is transmitted through the sensory neuron via the dorsal root ganglion to the central nervous system, where the sensory neuron synapses with a relay neuron. In part B, roundworms respond to physical stimuli by changing the direction in which they swim. The recovery time of roundworms following habituation was investigated. 3 groups of 25 roundworms were kept in separate petri dishes. The roundworms were habituated to a physical stimulus by tapping the side of the petri dish at different time intervals. Group 1 were habituated with 60 taps at 2-second intervals. Group 2 were habituated with 60 taps at 10-second intervals. And Group 3 were habituated with 60 taps at 60-second intervals. So the interval time is longer here in comparison to that. They say at 0, 0.5, 10, and 30 minutes after this habituation, each petri dish was tapped once and the number of roundworms whose swimming direction change was recorded. The graph shows the results of this investigation. So on the vertical axis, we have the number of roundworms changing direction, and on the horizontal axis, we have time after habituation. So it means this investigation was carried out after all the roundworms were habituated. So at this point where the time is 0 or 0 minutes, all the roundworms are habituated and therefore none has recovered. But we see after the time of habituation, there is increase in recovery because remember, on the vertical axis, we have the number of roundworms changing direction, meaning those are recovering from being habituated. So those in group 1 that were tapped with shorter taps showed greater recovery in comparison to all the others, and those in group 3 whose taps were longer, meaning those that were 60-second intervals, they were slower in recovery. We also see for all groups between 0 to 10 minutes the recovery is steeper or faster, and then it slows down from 10 onwards. And by the time we reach about 30 minutes, we see they are still going through recovery, meaning some of the organisms have not been recovered because there is a continuation, it has not leveled off. So they say calculate the percentage difference between the number of roundworms in group 1 and group 2 that change direction 30 minutes after habituation. So if we come to the graph, we can see in group 1 there were 20, the corresponding number is 20, and in group 2 the corresponding number is 12. So the percentage difference is going to be the difference, which is 20 minus 12, divided by the mean of the two values. The mean is going to be 20 plus 12 divided by 2, and then you multiply by 100. This gave me 50%. So 50% is going to be the answer. The next essay describes 3 conclusions about recovery times after habituation. This is similar to what I've explained on the graph. So here I said all experimental groups showed ability to recover after 0.5 minutes. Recovery is rapid between 0.5 minutes to 10 minutes and it's linear. However, after 10 minutes, recovery slows down for all groups. Organisms that had a shorter interval of habituation, for example, group 1, showed the fastest recovery time. And at the end of the test time, which is 30 minutes, organisms still showed increase in recovery, indicating that some are still recovering. So this brings us to the end of question 3. Let's continue to question 4. In question 4, the nervous system of an organism enables it to respond to a stimulus. Nociceptors and thermoreceptors are receptors that are sensitive to changes in temperature. An investigation compared the effect of temperature on the activity of thermoreceptors and nociceptors in the skin of volunteers. The graph shows the number of action potentials produced when each type of receptor was stimulated at different temperatures. So looking at the graph, on the vertical axis we have the number of action potentials per second, and on the horizontal axis we have the temperature. We can see thermoreceptors have more action potentials at lower temperatures in comparison to nociceptors. Nociceptors have zero action potentials between 34 to 42 degrees. They begin to show activity or action potentials after 42 degrees, but their numbers of action potentials at higher temperatures are more in comparison to the thermoreceptors. At this temperature, they have the same number of action potentials. So basically, you could say these show action potentials earlier, these have nothing until 42, but they increase and go beyond. At higher temperatures, you have more. These are kind of leveling off as those are still increasing. So they say, what is the ratio of the action potential produced at 48 degrees Celsius by nociceptors compared to thermoreceptors? At 48 degrees Celsius, here we have 13, and there we have 27. For nociceptors to thermoreceptors, it's going to be 27 to 13, which is 2.0769 to 1 or 2.1 to 1, so the answer should be D. And next they say nociceptors are better than thermoreceptors at preventing serious damage to tissues at high temperatures. They say comment on the extent to which the results of this investigation support this statement. This is true from the graph we saw. If I take you back, nociceptors are better at detecting changes, or they show greater action potentials at higher temperatures in comparison to thermoreceptors. So, like I've already explained in the graph, I said from the graph, thermoreceptors showed an increase in the number of action potentials produced until 44 degrees Celsius. After 44 degrees Celsius, the number of action potentials does not increase. That is for the thermoreceptors. And then nociceptors produce action potentials after 42 degrees Celsius. They produce larger increases in the numbers of action potentials per change in temperature, meaning they show a steeper gradient or steeper gradients compared to thermoreceptors. Although thermoreceptors showed sensitivity over a wider range of temperature or wider range of temperatures, however, there is no information about the ages of the volunteers, the gender, the sample size, and so on. Also, there is no information about repeat experiments and means, so we cannot conclude on the validity of the data. And again, remember this question is about commenting, so when you're answering commenting questions, you have to talk about the methodologies used, the validity of the methodologies. Then you talk about the sample size, you talk about the error bars if there is overlap or repeat experiments and so on. So that should be what you could write. In part B, scientists used 3 groups of mice, there is A, B, and C, to investigate the effect of nicotine on the transmission of nerve impulses. So in the first group, they said group A were given nicotine, group B were not given nicotine, group C were given nicotine but were deficient in the gene that produces the acetylcholine receptor on the postsynaptic membrane. Remember, nicotine binds onto those receptors, so if the genes that produce those receptors are missing, it means the receptors will not be there. So in group C, even if nicotine is there, there will be no sensitivity because nicotine has nothing to bind onto. So they said the mice in group A and C were given the same dose of nicotine over the same time period. The results are shown in the graph. On the vertical axis here, we have the number of action potentials in the postsynaptic neuron. And on the horizontal axis, we have the time, and nicotine was given here. We can see after nicotine is given, the number of action potentials increase greatly for group A And this could be because group A was given nicotine and they have acetylcholine receptors, so there is greater action potentials. But in group B, we do not have nicotine, so even though the acetylcholine receptors are going to be there, there is no increase in the number of the action potentials. In group C, nicotine is provided, but there are no acetylcholine receptors. That means there is no change in the sensitivity, and therefore the number of action potentials does not change. So we see group B and C have similar action potentials, while group A, there was a greater increase. So they say explain the results of this investigation. They want to use your own knowledge and the results in the graph to support your answer. So like I've already explained, from the graph, presence of nicotine causes an increase in the number of action potentials produced in the postsynaptic neuron because nicotine binds to the acetylcholine receptors, causing action potentials. Groups B and C have similar numbers of action potentials. Group B is not given nicotine, while Group C has no acetylcholine receptor producing genes, so it lacks the receptor. Even in presence of nicotine, the number of action potentials remain unchanged. So this brings us to the end of question 4. Let's continue to question 5. In question 5, small variations in DNA sequences and activation of genes can explain why individuals respond differently to disease and the drugs used to treat them. Describe how active genes could be identified. Now, remember, when genes are activated, messenger RNA is going to be produced. So messenger RNA is going to be detected because it shows us which genes are activated. And then we can use that to produce complementary DNA. The complementary DNA can be used with fluorescent dyes and probes in microarray, and we can be able to know which genes are being activated. We can also use bioinformatics to be able to interpret the results. The next they say epigenetic modifications can have an effect on the activity of all genes in cells, which is the role of histones in the cells. Histones are proteins onto which DNA wraps, so they give structure and support to DNA molecules, and therefore the answer should be C. And next they say which is the process that regulates gene expression. This is going to be epigenetic changes or epigenetic control. Crossing over occurs in meiosis. Post-transcription changes, these occur on messenger RNA. And post-translational changes, these occur on proteins. So the answer should be B. In part C, they say Prader-Willi syndrome is an inherited genetic disorder that involves changes on chromosome 15. The chromosome inherited from the father has genetic deletions, and the chromosome inherited from the mother has DNA methylated bases. Remember, when DNA is methylated, it's not going to be transcribed, and when the genes are deleted, it means they're not going to be converted into messenger RNA, and therefore the proteins are not going to be produced. So they say the diagram shows these chromosomes for an individual with Prader-Willi syndrome. These are the chromosomes. So like I already said, if there is methylation, transcription is going to be prevented, and if there are genes that are missing, there will be nothing to transcribe, and therefore no proteins are going to be formed. So this explains why these changes in the chromosome of this individual would cause Prader-Willi syndrome. Like I've already said, addition of methyl groups to the cytosine bases prevents transcription, and the regions with genetic deletions have no base sequence to be copied. So in both cases, RNA polymerase will not bind, or the binding of RNA polymerase will not occur. Messenger RNA will not be produced, and so protein synthesis will not occur. So next they say the symptoms of Prader-Willi syndrome can be treated using human growth hormone. This hormone is a protein produced by the pituitary gland. Describe how E. coli bacteria can be used to produce human growth hormone. So here we'll try to isolate the human growth hormone gene. This is going to be by using restriction endonucleases to cut the gene out of the human DNA. We'll also use the same restriction enzyme to cut a specific plasmid so that complementary sticky ends are left. And then we can use DNA ligase to insert the gene into the plasmid. After the plasmid can be introduced into E. coli, or the strain that is going to be used, and then we can use industrial fermenters at optimum conditions to culture, and the human growth hormone is going to be produced into the culture medium. It can be isolated and then purified. So this brings us to the end of question 5. Let's continue to question 6. In question 6, the fastest recorded speed by a human was set by Usain Bolt sprinting in 2009. He achieved a speed of 12.3 meters per second. Name the main type of muscle fiber that is being used in the leg of muscles of a sprinter. These are fast-twitch muscle fibers. Next, they say during a sprint race, leg muscles produce lactate. Describe what happens to the lactate at the end of the race. The lactate is going to diffuse into the bloodstream. It's going to be carried into the liver, and then it's going to be converted into individual components, like in this case. The liver is going to convert the lactate into glucose, and this glucose could be converted into glycogen, or this could also be converted into pyruvate, and the pyruvate can be used in the aerobic respiration processes. So I said lactate diffuses out of the muscle to the blood. It can be transported to the liver where lactate is oxidized to pyruvate. The pyruvate can be used in the link reactions during aerobic respiration. Also, the produced pyruvate can be converted into glucose or glycogen. Next, they say which of the following statements are features of muscle fibers in a long-distance runner. They say have reduced blood capillary networks, that is no. They have large numbers of mitochondria, that is correct. Contain a low concentration of myoglobin, no. So it's only 2. So the answer is going to be B. And next, I say which of the following explains how muscles contract. So the first one is Fick's law. This is for diffusion. The fluid mosaic model, this is the cell membrane. Lock and key model, enzyme substrate. A muscle contraction can be explained using the sliding filament theory or model. So the answer is D. In part B, exercise has many benefits for the health and well-being of an individual. An investigation was carried out on non-athletes and athletes. All participants did a series of physical exercises and several measurements were taken. The means of the measurements are given in the table. So we have the means for the resting heart rate. We can see it's higher in non-athletes than athletes. The peak heart rate, it's higher in non-athletes than athletes, but the values are close. The maximum volume of oxygen used during anaerobic respiration, it's lower here and higher among athletes. And then here, the maximum volume of oxygen used under aerobic respiration, it's higher here in comparison to here. The differences we see here, for example, this is going to be because the athletes have more muscle build or they carry out more respiration, so they will need more oxygen. In a different investigation, the relationship between power generated and oxygen use was studied in athletes and in non-athletes. So they say the results are shown in the graph. On the vertical axis, we have oxygen used. On the horizontal axis, we have power generated. For both, we see oxygen used increases as power generated increases. Since the values corresponding to the non-athletes stop about here, this is the maximum, we see athletes have more power in comparison to the non-athletes. And also, athletes use more oxygen. If I draw a vertical line, we can see beyond these values here, there is no power generation for the non-athletes. So athletes have more power generated. And also up here, we can see those values here. There is no result for the non-athletes. So here they say discuss the differences in the results for the athletes and non-athletes in these two investigations. Use your knowledge and information in the table and graph to support and explain your answer. So here I discussed the table first and then I went to the graph. So from the table, compared to non-athletes, athletes have higher maximum volume of oxygen used under aerobic and anaerobic conditions. And this is because athletes have more muscle cells that require more oxygen for aerobic respiration. Compared to athletes, non-athletes have higher resting and peak heart rates because their heart muscle needs to contract more to deliver oxygen to the tissues. From the graph, as the power generated increases, oxygen used by both athletes and non-athletes increases. Athletes use more oxygen than non-athletes. Also, more power is generated by athletes than non-athletes. Athletes do more training, so they have more muscle development. Their lungs and hearts are adapted for efficiency, and they use more oxygen for aerobic respiration. They contain more mitochondria for ATP required in muscle contraction. From the table, mean values are used, but no standard deviation, so we can't comment on the validity of the results. So this brings us to the end of question 6. Let's continue to question 7. In question 7, the kangaroo rat is a mammal adapted to living in a warm, dry habitat. The resting body temperature of a kangaroo rat is 37 degrees Celsius. During exercise, the body temperature is maintained between 38.5 to 40 degrees Celsius. Explain how the body temperature can be regulated to stay between 38.5 to 40 degrees Celsius. This involves thermoreceptors as well as the thermoregulatory center, depending on whether the body temperature is higher or lower. If it's higher, then the metabolic rate has to decrease, meaning impulses are going to be sent to the liver to decrease metabolism. Also, impulses will be sent to the skin for vasodilation to occur. But if the temperatures are lower, then more metabolism is going to be carried out to generate more heat to increase the body temperature, and then vasoconstriction will occur to minimize heat loss. So I say thermoreceptors detect the changes in the temperature. They send impulses to the thermoregulatory center, which sends impulses through the motor neuron to the effector organs, for example, the skin or liver, to carry out negative feedback. If the temperature is high, vasodilation in the skin occurs, the hair erector muscles relax, the liver slows down metabolism to decrease heat generation, and these work together to decrease the temperature. In part B, in mammals, urine is produced by ultrafiltration and the selective reabsorption of solutes and water. Kangaroo rats adapted to survive on a diet of dry seeds and little or no drinking water. Suggest how kangaroo rats adapted to survive in areas with little drinking water and dry seeds. Survival could be behavior or physiological. So first, they alter their behavior by hiding underground when it's hot in order to cool down to prevent excessive water loss. They also have nephrons with longer loops of Henle, so more sodium ions are pumped from the ascending limb of the loop of Henle, and this increases the ion concentration of the tissue fluid, so more water is going to be reabsorbed. In part C, they say the diagram compares the daily water balance between a kangaroo rat and a human. So here we see water balance in a kangaroo rat. This is 2 centimeters cubed per day, and for the humans is 2,500 centimeters cubed per day. When we look at the water gained, most of it is going to be gained from metabolism in the kangaroo rat, and little is going to be gained from food, meaning they use a higher percentage of metabolic water. For the humans, the greatest percentage is going to be gained through liquids, and then next is going to be food, and the least is going to be metabolic water. Humans gain most from liquids because they have access to drinking water, while kangaroo rats that live in deserts have no access to drinking water, or there is less access to drinking water. Then water loss. Here we see the greatest percentage is going to be by evaporation, and again they stay in a desert, so the temperatures are really hot, and the least is going to be through feces. For the humans, the greatest is going to be through urine, and again there is more water that they can drink. Least is going to be through feces, while evaporation also has quite a bit of a percentage. Next, they say calculate the difference in urine produced over 7 days by the kangaroo rat and by the human. They want you to give your answer in standard form. So we saw from the table humans produce 1,500 per day, and in a week it's going to be 1,500 times 7, which gives us that volume. The kangaroo rat produces 0.45. If I take you back, humans have the urine, 1,500, and the kangaroo is going to be that. So for the kangaroo, it's going to be 0.45 per day times 7, 3.15. And then the difference is going to be that minus that, giving us this. To write that as standard form, it's going to be taking 1, 2, 3, and 4. So it's 1.049685 times 10 power 4. And rounding off to 3 significant figures, it's 1.05 times 10 power 4. That's my answer. And next is a comment on the differences in the water gain and water loss between the kangaroo rat and the human. Use your knowledge and information in the diagram to support and explain your answer. I've already gone through the results when we were looking at the table, so I'm just going to read what I wrote. I said humans have higher water balance, which is 2,500 centimeters cubed per day, than kangaroo rats that have 2 centimeters cubed per day. Kangaroo rats gain most water from metabolism, which is that, while humans gain most water from liquid. Kangaroo rats live in a desert, so there is less water to drink. The humans lose most water as a percentage through urine, while kangaroo rats lose most water as a percentage through evaporation. And this is because kangaroo rats live in the desert where temperatures are high. Also, kangaroo rats have longer loops of Henle, so they produce concentrated urine. For both, the net water gain is 0 because the osmotic potential has to be maintained, meaning homeostasis has to be carried out. So this brings us to the end of question 7. Let's continue to question 8. Now, question 8, we begin by looking at the scientific article. So I'm just going to be quiet as you guys observe this. And then I'll go back to the other question. In question 8, they say the scientific article you have studied is adapted from an article in Clinical Nutrition entitled Muscle Tissue Changes with Aging. It was published in July 2004. They say use the information from the scientific article and your own knowledge to answer the following questions. First question says give a definition of the term sarcopenia. Sarcopenia is a decrease in muscle mass and function which is associated with aging. And in part B, they say satellite cells are stem cells. This is from paragraph 2. Explain how a specialized cell, such as a muscle cell, is produced from a stem cell. This is because gene expression is going to be carried out. Some genes are going to be switched off and others are going to be switched on. Certain proteins are going to be produced and these could control the functioning of the cell. So I said epigenetic modification occurs. Transcription factors cause expression of some genes. Messenger RNA is going to be produced. And messenger RNA is going to be translated into proteins like actin and myosin. Remember, these are proteins within the muscle. And these are going to change the structure and function of the stem cells, becoming a muscle cell. Next, they said testosterone is a steroid hormone. Describe how reduced production of testosterone in the male testes can result in reduced muscle protein. From the paragraph parts we read, I'm going to try to expand this out. I copied it. So here they say since testosterone increases the muscle protein synthesis, muscle mass and strength. This means presence of testosterone leads to increase in muscle strength, muscle mass and so on. Since testosterone is a hormone, it could act as a transcription factor, or it's part of what acts as a transcription factor, causing the expression of certain genes and therefore the formation of certain proteins within the muscle. So I say testosterone is a steroid hormone, so it can enter the muscle cell through the cell membrane. If less testosterone is present, less will cross the muscle cell membrane to bind to receptors. And fewer receptor testosterone complexes will be formed, and then fewer complexes will act as transcription factors. Less transcription will occur, and less translation will occur, meaning fewer muscle proteins will be produced. In part D, they say insulin control of blood glucose is an example of a negative feedback loop. Explain how a negative feedback can maintain a system within a narrow limit. For negative feedback to work, we need receptors or things that detect the changes. They are going to stimulate certain molecules to be produced that control homeostasis. So I said there are receptor cells to detect changes, and the receptors then stimulate the effector organs to carry out a response which offsets the changes. For example, the chemoreceptors detect a change in glucose concentration, and again here the pancreas is stimulated to release insulin if it's against an increase, or glucagon if it's against a decrease. So if insulin is produced, then it's going to convert glucose to glycogen. Next they say, suggest how insulin increases the uptake of glucose by muscle cells. This is going to be by facilitated diffusion because muscle cells or any cells take up glucose through facilitated diffusion. So for this to occur efficiently, we need more channel proteins in order for more glucose to be taken up. So I said insulin binds to the receptor of muscle cells, and this leads to the production of more glucose transport molecules in the cell membrane. So more glucose is going to be taken up by facilitated diffusion. Aerobic exercise may increase muscle size in older individuals. Describe how computed tomography scans can be used to measure these increases in muscle size in older individuals. CT scans use X-rays, and this gives a cross-section image. These scans can be taken before and after aerobic exercise, and then the images can be compared to see if there was an increase or not. Next is explain why aerobic exercise increases mitochondrial density and activity. That is from paragraph 9. For this, you need to go back and read, but we're going to focus on the mitochondrial density. If there is increase in energy demands like during exercise, there is going to be an increase in the replication of more mitochondrial DNA. This will lead to a production of more mitochondrial proteins, and the mitochondria produced are going to be more efficient, meaning there is going to be increase in density as well as activity. As I said, mitochondria are more active during more aerobic respiration, meaning their efficiency is improved, so more aerobic respiration occurs and more ATP is synthesized. ATP is useful in active transport of calcium ions and for breaking the actomyosin cross bridges during muscle contraction, so more mitochondria are produced, increasing the density. This means activity is going to increase and the density increases. And lastly, they say whey and egg protein are often used to supplement protein-poor diets. Explain why diets rich in these proteins may not be ideal for prolonged treatment of sarcopenia, and they're referring to paragraph 11. If I make paragraph 11 expanded out for you to see, they say non-essential amino acids comprise of significant portions of dietary proteins, including the high-quality proteins like whey and eggs. It means these contain non-essential amino acids, and these may not be important or necessary in the stimulation of muscle growth. So I said because whey and egg protein contain non-essential amino acids, these are not necessary for acute stimulation of muscle protein anabolism. Anabolism is making the muscle protein among older people, but it could provide huge amounts of calories. And these calories provided could be stored as fat instead of being muscle. So I went on to say these high energy molecules could be converted into fat without an increase in muscle mass, so they are not a suitable solution for sarcopenia. So this brings us to the end of question 8 as well as to the end of the whole paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.